Let's talk bacteriology. You know it's a huge topic for step one, right? So we're going to zero in on the stuff that's super high yield, basically how these bacteria actually make us sick.
So let's just jump right in. The best way to think about all these different bacterial mechanisms is to see them as a full-on arsenal. We're talking tools for invasion, for survival, for attack. It's not just a list of random facts you have to memorize. It's a playbook.
Okay, so first things first, let's look at their defensive gear. I mean, before a bacterium can even think about launching an attack, it's got to survive our body's own military, the immune system. And they have some pretty clever ways of doing that. You know, some bacteria are like the ultimate survivalists. When things get tough, they form these things called spores. Think bacillus, think clostridium. These spores are, well, they're incredibly tough, super resistant. And this is why autoclaving, at 121 degrees Celsius is always going to be the classic exam answer for sterilization. Yeah, sure, other things work, but for the boards, it's all about the autoclave. All right, now let's talk about their active defenses. So you've got these polysaccharide capsules that are basically a slippery shield, makes it hard for our immune cells to grab on. Then you have protein A from good old Staph aureus, which does this really clever thing where it binds antibodies backward, making them totally useless. But the one you absolutely have to circle in your notes is M protein from group A strep. It does two things. One, it stops phagocytosis, but two, and this is the kicker, it mimics our own cardiac myosin. This whole strategy, we call it molecular mimicry, is the reason it can lead to that autoimmune attack on the heart in acute rheumatic fever. That is a classic exam trap. You gotta know that. Okay, so where do bacteria get all these new weapons and shields from? Well, they share. It's like they're passing notes in class. This process is called horizontal gene transfer, and let me tell you, it is a huge topic for your boards. Why? Because this is pretty much the main way antibiotic resistance spreads. So, first up, we have transformation. You can think of this as bacterial scavenging. One bacterium dies, splits open, and its DNA spills out everywhere. Then, another competent bacterium just comes along, scoops up those naked DNA fragments, and integrates them into its own genome. The big players you need to remember for this are strep pneumo, H. flu, and Neisseria. Then there's conjugation. This one's a lot more direct. It's basically a gene swap through a little bridge called a sex pilus. And yeah, this is a major route for spreading those nasty antibiotic resistance genes. And then we have transduction, which is it's a brilliant little mechanism. You have a bacteriophage, a virus, that basically acts like a mistaken delivery service. In its simplest form, which we call generalized transduction, the phage just accidentally packages any old random piece of bacterial DNA. But there's a more specific version, and this is the one you absolutely have to know for your exam. And that is specialized transduction. Here, it's not random. It's when only specific genes, the ones sitting right next to where the phage has plugged itself into the bacterial chromosome, get transferred. So the bottom line for your exam, you have to know which toxins are passed around this way. And the best way to do that is with this mnemonic, ABCDS. Just burn it into your brain. A for group A strep's erythrogenic toxin, B for botulinum, C for cholera, D for diphtheria, and S for Shiga toxin. Seriously, if you see any of these toxins in a question stem, your brain should immediately scream specialized transduction. Okay, let's move on to the main event, the offensive weapons. And listen, you absolutely, positively must know the difference between exotoxins and endotoxins. I mean, know it cold. This is pretty much guaranteed to show up on your exam in some form. Think of this table as your ultimate cheat sheet. So exotoxins, they're proteins. They're actively secreted by both gram-positives and gram-negatives. They are highly toxic, very specific in what they do, and, super important, we can make vaccines against them. We call those toxoids. Now, endotoxin is a different beast entirely. It's only in the outer membrane of gram-negative bacteria. It's a lipopolysaccharide, or LPS for short. It only gets released when the cell dies and breaks apart. It's actually less toxic, but it's famous for causing fever and shock. And no vaccine for this one. And this image really makes it crystal clear, right? The exotoxin is like a guided missile being actively launched from the bacterium to hit a specific target. The endotoxin, though, is more like the shrapnel from the bacterium itself. It's part of its structure, and it only causes damage when the whole thing blows up. All right, let's do a quick rapid-fire round. We're going to go through some of the most clinically important toxins and how they work. Pay attention here, because these are the kinds of details that separate the good scores from the truly great scores. Okay, you really need to focus on this one. Superantigens, like the infamous toxic shock syndrome toxin from Staph aureus. These things are totally unique. They don't care about specific antigens. Instead, they act like a master key, or maybe like glue. They just non-specifically lock T cells to antigen-presenting cells. And what happens next? A massive, uncontrolled T cell activation, what we call a cytokine storm. And that is what directly causes the crazy fever, the rash, and the severe hypotension you see in toxic shock syndrome.
Now, for those E. coli toxins that give you that gnarly, watery diarrhea, there's a classic mnemonic you've got to know. The heat-label toxin increases adenylate cyclase. So think label in the air. And the heat-stable toxin increases guanylate cyclase, so stable on the ground. Both of these pathways, CAMP and CGMP, end up causing electrolytes to be secreted into the gut, water follows, and bam, watery diarrhea. So let's circle back to endotoxin just for a second, because this is so important. This flowchart really shows the whole cascade. So the truly toxic part of LPS is this molecule called lipid A. When it gets released into the bloodstream, it's like a bomb going off. It triggers macrophages, it triggers the complement system, and it triggers the coagulation cascade, all at the same time. It's this insane domino effect that leads directly to the classic signs of septic shock: fever, crazy low blood pressure, and that dreaded disseminated intravascular coagulation, or DIC. And hey, if you want to remember that whole storm of effects from endotoxin, this mnemonic right here is going to be your best friend. It walks you through everything: edema, nitric oxide release that causes the hypotension, DIC, the key cytokines like TNF-alpha, IL-1, and IL-6, all the way to the final outcome, shock. All right, so theory is one thing, but you know the USMLE is all about application. We've gone over the bacterial arsenal. Now let's see if you can put it all together and diagnose the aftermath with a couple of super high-yield clinical cases. Okay, let's break this one down. What are the clues? We've got a patient in shock. They have a diffuse, sunburn-like rash. That is a huge buzzword. And you've got the perfect setup for a Staph aureus infection: recent nasal packing. So what kind of toxin could cause this massive systemic syndrome? And if your brain went straight to superantigen, you are 100% correct. This is the absolute textbook presentation of toxic shock syndrome. And remember, the TSST1 superantigen is so potent, you don't even need a big, widespread infection. All it takes is a localized colony of staph, just pumping out this toxin that gets into the bloodstream and kicks off that devastating cytokine storm. Okay, next case. Let's look for the buzzwords again. We have an infant, they're constipated, and they have floppy baby syndrome, which is just classic USMLE speak for descending flaccid paralysis. But the biggest clue, the one that should make a light bulb go off, is the raw honey. So what's hiding in that honey that a baby's immature gut just can't handle? The answer has to be botulinum toxin. This is probably the highest yield association you can possibly learn for infant botulism. The baby ingests the spores from the honey, those spores germinate in their gut, and then they start producing the toxin. And what does that toxin do? It gets to the neuromuscular junction and blocks the release of acetylcholine. No acetylcholine, no muscle contraction. And that's what leads to that classic flaccid paralysis. So what's the big takeaway here? It's that all these mechanisms, they're not just facts to memorize. They are the actual playbook for how bacterial infections work. And if you understand the playbook, you can understand the diagnosis and the treatment. So you've decoded it. The real question is, what will you diagnose next?